Dear viewers, I am Dr. Nitu Debnath. I have prepared this presentation in response to the great initiative taken by the Zoological Society of Assam to facilitate the learning processes of both UG and PG students of zoology in Assam. Today I am going to discuss about the fundamental functional property of nervous system, and that comprises of the generation and propagation of nerve action potential. I am dedicating this presentation to my revered teachers who taught me physiology at Guwahati University. The learning objectives of this presentation will include the understanding of basic structure of neuron, the concept of resting membrane potential, the concept of nerve action potential, the understanding of the generation of nerve action potential, and finally we'll also include the mechanism of propagation of action potential through the two important types of nerve fiber, the myelinated and non-myelinated nerve fiber. Now let us start our discussion with the basic structure of neuron. As you know from earlier classes also, that a neuron or nerve cell is comprising of two main parts. One is the cell body containing the nucleus at the center, and the other part is known as the axon. The cell body, from the cell body of neuron, there are some branching which arises, and these branches are known as the dendrites. There may be 5 to 7 dendrites in a particular cell body, and from this cell body, the axon, it also extends on the other direction, and this axon terminates in some very special structures known as the synaptic button or axon telodendria. And in case of the myelinated neurons, the axon intermittently is covered up with the myelin sheath formed by the Schwann cells. So in mammalian neuron, there may be 5 to 7 dendrites which extends from the cell body, and a long fibrous axon which originates from axon hillock. So axon hillock is that part of the cell body from which the axon originates, and this is a very special part of the cell of the neuron where there are many functions of this axon hillock in signal transmission, in signal computation, etc., which I'll discuss in some other presentation. Now we are going to discuss the differences between the myelinated and non-myelinated neuron. In case of myelinated neuron, as I told, that there are myelin sheaths around the axon, and these are formed by the Schwann cells. But in case of the non-myelinated axon, there are no such myelin sheath, and axon is devoid of such myelin sheath or Schwann cell. The myelinated neurons are mainly found in the mammalian neuron and all central nervous system neurons. But non-myelinated neurons, on the other hand, are found in invertebrates, and where there are no Schwann cell, and as a result of that, the wrapping of the Schwann cell membrane is not there in the non-myelinated axon. If we look close at this diagram, we can understand the association of the Schwann cell and the axon. Here, in the cross section of the axon, it is seen that the axon is at the center, and there are some protein lipid complex which is formed by the Schwann cell. This is the nucleus of the Schwann cell, and the plasma membrane of the Schwann cell wraps around the axon many thousand times. And as a result of that, a protein lipid complex is formed around the axon, and that is nothing but the myelin sheath in case of the myelinated neuron. So, as I told, this is a protein lipid complex, and it is formed by the wrapping of the Schwann cell plasma membrane. So Schwann cells are basically glia-like cells, but in case of the mammalian central nervous system, this myelin sheath is actually produced by the oligodendrogliocytes. But outside the central nervous system, Schwann cells form the myelin sheath. So in case of myelinated neurons, the myelin sheaths or fragments of myelin sheaths are formed by the neighboring Schwann cells present on the axon. But there are some 1 micrometer constrictions between two fragments of myelin sheaths, and these spaces where the axon is exposed and not covered up is known as the node of Ranvier. So far as the size is concerned, the mammalian axons are 20 micrometer in diameter, while the giant unmyelinated neuron in invertebrates may be 1 micrometer in diameter. Here we can see that the neuron to neuron connection or synapse. Here, this is one neuron and this is another neuron having the cell body and the axon. Here, this is the cell body and this is the axon. Now, these two neurons are connected at these points where the axon of this cell is making some special connection with the dendritic ends of the neighboring cell. And these special connections are known as the synapse. And here it is also showing the signal direction. That means when the action potential is generated or impulse is generated in one neuron, it is transmitted in this direction from the axonic end to the dendrite. And from this cell body, again, it will go to the axon and to the neighboring neuron through the synapse. Now let us come to the second learning objectives, the concept of resting membrane potential. So the membrane potential is the ability of cell membrane to maintain an unequal distribution of ions in and outside of the cell. In this table, here the concentration of potassium, sodium, and chloride ions, it is shown that in intracellular concentration and extracellular concentration, that is the outside of the cell and inside of the cell concentrations are differing. And for potassium ion, intracellular concentration is higher than the outside. For sodium, the extracellular concentration is higher than the inside. Same with the chloride ions. This unequal distribution of ions creates a potential difference between inside and outside of the membrane. And this potential difference developed due to the membrane characteristics is called the membrane potential. So the potential difference that exists across the plasma membrane, it exists when the cell is at rest, and that's why membrane potential is also called the resting membrane potential. This resting membrane potential varies from cell to cell and species to species. In case of mammalian muscle cell, the resting membrane potential may range from minus 80 to minus 90 millivolt, where minus 86 millivolt is contributed only by the leaky potassium ion channels, while only minus 4 millivolt is contributed by sodium-potassium pump. The small muscle fiber, which is present in the sino-auricular node in the right atrium, may have a resting membrane potential below minus 70, and it may range from minus 55 to minus 60 millivolt, because these fibers are spontaneously leaky to sodium ions. While the nerve cells resting membrane potential may range from minus 70 millivolt to minus 90 millivolt. So this concept of membrane potential or resting membrane potential, let us understand how the membrane potential is measured. The membrane potential is measured by inserting microelectrode inside the plasma membrane with a reference electrode just outside the plasma membrane. The plasma membrane, as shown in this diagram, is having the lipid bilayer, and just inside of the plasma membrane, that is the cytosolic part, is negatively charged, while the extracellular fluid part is positively charged. And at that situation, the plasma membrane is said to be polarized. So when the microelectrodes are connected to the voltmeter, the membrane potential is shown, or the potential difference is shown as negative value of minus 70 millivolt or minus 90 millivolt. This minus or negativity is because the membrane potential is always measured with respect to the inside of the membrane. And since the inside of the membrane is negative, so the resting membrane potential is also negative. So for any cell or for a neuron or part of neuron, we can measure the resting membrane potential. So the concept of membrane potential and resting membrane potential, let us discuss the factors responsible for unequal distribution of ions across membrane, which is mainly responsible for the membrane potential or resting membrane potential. The first factor responsible is the diffusion potential, and that diffusion potential is dependent again on the concentration gradient of various ions. So concentration gradient means concentration slope. For example, for the potassium ion, the concentration slope is from the inside to the outside, because the concentration of potassium ion is higher inside than the outside. And for the sodium ion, the gradient is just the reverse, from the outside to the inside, because the sodium ion concentration is higher outside the cell at resting state. The second factor responsible is the electrical gradient and Donan's equilibrium. To understand it, let us discuss a little bit more about the Donan's equilibrium. American physicist Joshua Wheeler Gibbs in 1878 and British chemist Frederick G. Donan in 1911 have actually experimentally proved that there exists an equilibrium between the distribution of ions and molecules in and outside of the living cell. And they call this phenomenon as the Donan's effect or the Gibbs-Donan effect. And this, this effect is due to the permeability characteristics of the plasma membrane, because we know that the living cell plasma membrane is actually having the characteristics of semi-permeability, or sometimes we call the selectively permeable membrane. And because of that, it can allow some of the ions and molecules to pass through it, while others cannot pass through it. So the protein molecules, along with the organic phosphates, which are negatively charged, they actually accumulate inside the cell, and they cannot cross the plasma membrane. While and this phenomenon is known as the Gibbs-Donan effect. On the other hand, the sodium ions, which are having lesser permeability or lower permeability than the other ions, and because of that, they remain higher concentration in outside, and that is contributed also by the sodium potassium pump, which continuously pumps sodium ion from inside to the outside. And because of the lower permeability of the sodium ion through the membrane, as well as the contribution by the sodium potassium pump, the concentration of sodium ion is higher outside, and this is also called the Donan's effect. And at the summary of these two effects of, or the Gibbs-Donan effect, they have explained that the sum of diffusible and non-diffusible anions is equal to the sum of the diffusible and non-diffusible cations on either side of the membrane. And this is known as the Donan's equilibrium. And this Donan's equilibrium ultimately leads to the unequal distribution of diffusible ions, and that ultimately leads to the potential difference across membrane when the cell is at rest. And here the Gibbs-Donan equilibrium is represented in this diagram. You can see that the sodium ion concentration is higher outside than the inside, while the potassium ion concentration is higher inside than the outside. The chloride ion concentration is higher outside than the inside. But in the cytosolic or intercellular part, there is the presence of negatively charged protein. And at one time, at resting stage, the sum total of the anionic groups and sum total of the cationic groups on either side of the membranes are equal. And this is known as the Gibbs-Donan equilibrium. The third factor responsible for unequal distribution of ions is the electrogenic pump or sodium potassium pump, which is an ATP dependent protein channel which can transport sodium ions and potassium ions against their concentration gradient, and that's why they contribute to the unequal distribution of ions. The fourth factor is the presence of sodium potassium leak channels, and through which the sodium and potassium can easily leak through the plasma membrane from inside to the outside or outside to the inside according to their concentration gradient. So here are some facts regarding the permeability of potassium and sodium ion across membrane. The permeability of potassium ion is higher than the sodium ion. Sodium potassium leak channels are more permeable to potassium ion, almost 100 times more than the sodium ions. And the reason for that is that the potassium ion binds fewer number of water molecule, and so in hydrated condition the size of potassium ion is smaller than the sodium ion. Sodium ion can bind 4.5 water molecule. Excess of sodium ion blocks cellular enzymatic activities, and that's why when the action potential is generated in neuron, sodium ion already entered and they remain there, and if they remain for a long time they can impair the enzymatic activities, and that's why the sodium potassium pump plays a very important role in fluxing out the sodium ion which has already entered inside the cell to the outside, because this sodium potassium pump is an ATP dependent uh, protein channel which can pump the sodium ion and potassium ion against their concentration gradient. So with this understanding of the membrane potential, let us discuss the third and the most important learning objective of this presentation, that is the nerve action potential and how a nerve action potential is generated in neuron. So to understand the nerve action potential, let us discuss the successive stages of nerve action potential. This diagram represents one nerve action potential where in y-axis the millivolt membrane potential in millivolt is shown, and in the x-axis the time in millisecond is shown. So the first stage of nerve action potential is called the resting stage. At this stage, the resting membrane potential exists before the action potential occurs. At this resting stage, the membrane is said to be polarized and the membrane potential is large and negative, which may range from minus 70 millivolt to minus 90 millivolt with respect to the inside of the membrane. The second stage is known as the depolarization stage, when the membrane potential rises towards zero and up to plus 35 millivolt. The third stage is known as the repolarization stage, when the membrane potential again falls from plus 35 level and again become negative, or it reaches the minus 90 level. And the fourth stage is known as the hyperpolarization, when the membrane potential falls below the resting membrane potential. That is, if it goes below the minus 90 or minus 70 level, then that is known as hyperpolarization. If we look close at this diagram, we can find that the resting membrane potential exists at minus 90, and when the membrane potential goes above up to the minus 65 millivolt level, then the depolarization process continuously goes on from minus 90 to minus 65, and that happens because of the leaky sodium ion channels are open and a few sodium ions come inside the plasma membrane of a cell. And if the membrane potential reaches minus 65 because of the sodium ion entry, then the depolarization process rapidly goes towards the zero and up to the plus 35 level. So depolarization process, when it becomes rapid suddenly at minus 65 or minus 75 millivolt level, that level is known as the threshold, when the voltage gated sodium ion channels open and large number of sodium ions enter inside the cell, which leads to the rapid depolarization process. And when the membrane potential reaches to plus 35, again the potassium ion channels or the voltage gated potassium ion channels open, and then the membrane potential again comes down from plus 35 towards the resting membrane potential of minus 90 millivolt. At the final stage, when the membrane potential reaches minus 90, the the repolarization process still continues because sodium ions efflux from the cell to the outside continues for some time, and that leads to the hyperpolarization. We will discuss in detail about the depolarization and repolarization in the later slides. Here you can go a little further to understand the action potential in detail. And we are to understand that different stages of this action potential are achieved due to different ions, mainly the potassium and sodium ion, and the channels. Like in threshold, the threshold is a stage which is achieved due to the sodium leak channels. It may be also due to local potential change. And these sodium leak channels are very important channels, protein channels, through which the sodium ions can leak inside the cell. And they are responsible for this threshold or increase in membrane potential from minus 90 to minus 65 or maybe minus 75. Second stage is depolarization, and the depolarization is because of the voltage gated sodium channel. The voltage gated sodium channels are special channels which are voltage dependent. Simply means that the increase in voltage or the increase in membrane potential cause these sodium ion channels to open. The third stage or repolarization stage is because of the voltage gated potassium channels. And the fourth stage is also due to voltage gated potassium channels. We'll discuss the roles of these voltage gated potassium and sodium channels in the achievement of the action potential in a neuron. So let us discuss here the depolarization stage of the action potential. Here, any mechanical or chemical or electrical stimuli, if it is reached in the plasma membrane of a neuron, it causes sodium potassium link channels to open. And because of this opening, the increase in membrane potential up to plus 15 millivolt is achieved. And that is because of entry of a few sodium ions through these sodium potassium link channels. So this increase in membrane potential from minus 90 to minus 75 or minus 65, it causes a voltage change, and there are some very special channels like the voltage gated sodium ion channels which respond to this change in voltage, and sudden conformational change occurs, and as a result of that the sodium ion channels, voltage gated sodium ion channels open, and it leads to the increase in sodium ion permeability. Up to 5,000 fold increase in permeability occurs, and where 6,000 sodium ions can enter in a particular cell per second. So when the this stage is achieved, this is known as the threshold or firing level. And the oscilloscope tracing rapidly reaches and overshoots isopotential or zero potential and reach up to plus 35. Here we can discuss about the characteristics of the voltage gated sodium ion channel in detail. In this diagram, if you see here, this is the voltage gated sodium ion channel which is closed at resting stage, and the outside of the membrane is positively charged, inside is negative. That means the plasma membrane is polarized and the voltage gated sodium channels are closed. Characteristically, the voltage gated sodium ion channels are having two gates. One is called the activation gate and another is called the inactivation gate. So the change in membrane potential from minus 90 to minus 65 or minus 75, like that 15 millivolt increase in membrane potential, causes the opening of the activation gate because of the conformational change occurs in the protein, and that conformational change opens the activation gate, and as a result of that the sodium ion can rush inside the cell through this channel because sodium ion concentration is higher outside the cell than the inside. So there is a natural tendency of these ions to diffuse inside the cell, and when the sodium ions are entering rapidly, then the membrane potential goes from minus 65 to 0 and up to up to plus 35 level, and that brings about the depolarization stage. Now, when the membrane potential reaches to plus 35 level, the inactivation gate of the voltage gated sodium ion closes, and as a result of that the sodium ion entry stops, and depolarization process also stops at plus 35. Let us discuss some very special facts about this depolarization stage or the conformational changes which is occurring in these voltage gated sodium ion channels. So conformational change due to 15 millivolt increase in membrane potential opens the activation gate and also closes inactivation gate. That means the same conformational change which causes the activation gate to open also closes the inactivation gate. But this closing of inactivation gate is a very slow process, and before the closing of the inactivation gate, thousands of sodium ion enter inside the cell, and as a result of that the membrane potential goes to positive level and the depolarization is achieved. The sodium ion channel remains open for a few 10,000ths of a second and results in depolarization of nerve membrane due to rapid sodium ion entry. Now let us start discussing the second stage of the action potential, that is the repolarization stage. Here, the important ion channel which is responsible for this stage is the voltage gated potassium ion channel. Unlike the voltage gated sodium ion channel, the voltage gated potassium ion channels are having only one gate, and that is called the activation gate for the potassium ion. So this gate is actually remain closed when the membrane potential is at resting stage. That means when the minus 90 millivolt of membrane potential is there, this activation gate for potassium ion remains closed. But when the change in membrane potential from minus 90 to minus 65 or minus 55 or minus 75 occurs, that leads to the opening of the activation gate for potassium ion. But characteristically, the voltage gated potassium ion channels, they opens very slowly, slower than the closing of the inactivation gate for sodium ion. So here it is shown that at resting potential, it is closed and it opens when the membrane potential goes from minus 70 to plus 35. And it again closes very slowly from the plus 35 level to minus 80 millivolt when the hyperpolarization is achieved. Let us see the sequential events in the repolarization stage. The gate of potassium ion channel are usually closed at resting stage, but when the electrical or mechanical or chemical stimulus is reached in the membrane, there occurs sodium ion entry through the leak channels, and that leads to the increase in membrane potential to plus 15 millivolt. And that opens the potassium ion channel because there occurs a slow conformational change in the potassium ion channel. As a result of that, potassium ion diffuse outside from inside because potassium ion concentration is higher inside the cell than the outside. And there occurs a simultaneous decrease in sodium ion entry into the cell. So sodium ion entry decreases and the potassium ion exit increases at a stage. And these two stages ultimately brings the repolarization process. The fact remains is that the potassium ion channels open fully when the sodium channels or inactivation gate of the sodium channels are beginning to close. And when this potassium ion exit occurs and the sodium ion entry decreases, then the repolarization process it is speeds up and the recovery process of resting membrane potential occurs within 10,000ths of a second. The potassium ion channels begins to close at resting membrane potential. Even at resting membrane potential, the potassium ion gates remains open and potassium outflow continues because the fact is that the potassium ion channels they open slowly and they closes also slowly. So when the resting membrane potential is achieved, then also the potassium ion channels remain open and outflow of the potassium ion continues. And as a result of that, the small but prolonged hyperpolarization is achieved. So here we can summarize the repolarization stage. As we have already mentioned that repolarization process starts from plus 35 millivolt and it completes when the resting membrane potential is reached. So the most important ion channel, which is responsible for this repolarization process, is the voltage gated potassium ion channel. And characteristically, this potassium ion channel remains closed at resting stage or resting membrane potential. But when, because of any stimuli, the membrane potential reaches to minus 65 millivolt, then the threshold level is reached. And that threshold level, when it's reached, it leads to the opening of the potassium ion channel. So it opens slowly, and it completely opens when the membrane potential reaches to plus 35 level. And the exit of potassium ion occurs from inside to the outside, and as a result of that, membrane potential again goes down from plus 35 level towards the resting membrane potential. And because of the slowness of closing of the potassium ion channel, a fused potassium ion excessively goes out of the cell, and that leads to the hyperpolarization stage. Here you can summarize the generation of action potential. The resting membrane potential minus 90 millivolt. Some stimuli in the form of electrical or chemical stimuli, if it strikes the neuron membrane, then the sodium ion entry occurs through the leak channels and a threshold is achieved. And once the threshold is achieved, the voltage gated sodium ion channels open and a rush of the sodium ion occurs inside the cell. As a result of that, the depolarization process occurs and membrane potential reaches to plus 35 or plus 25. When it reaches plus 35, the repolarization starts because the voltage gated potassium ion channels open and the potassium exit occurs. As a result of that, membrane potential again goes to resting membrane, and the hyperpolarization stage is achieved because of the excess exit of the potassium ion channel because of slow closing of the voltage gated potassium ion channels. So an important part of this action potential is that there occurs a rapid depolarization process because of rapid entry of sodium ion, and at the same time, when the repolarization process occurs, the first stage is a very rapid process of repolarization. So a sharp rise and rapid fall of membrane potential is actually known as the spike potential of the axon. Here you can discuss an important aspect of generation of action potential, and that is all or none law. It states that a minimum intensity of stimulus is required to produce the action potential in nerve fiber. This minimum intensity of stimulus is known as threshold, and for nerve fiber, it may be minus 55 millivolt or minus 65 millivolt. It simply means that if the threshold is achieved, then action potential will be generated. But if the threshold is not achieved, action potential is not generated. Moreover, if the intensity of threshold is above minus 65 or minus 55, that does not change the amplitude or form of the action potential. And a sub-threshold magnitude of stimulus fails to generate the action potential. Once the action potential is generated at any one point of a neuron, it can excite the entire cell. This is the diagram of the neuron to neuron connection where the two neurons are connected through the synapse. Here, we can explain the all or none law by explaining that if a neuron is excited or if certain stimulus is reached in the plasma membrane of this neuron, then it can generate action potential, and this action potential will spread across the axonic membrane, and finally it reaches the synapse, and from synapse this impulse or action potential is transmitted to the neighboring cell, and it goes again from the cell body to the axon. And if the action potential is not generated here in this neuron, then it cannot spread and it cannot reach the synapse, and it cannot also be transmitted in the neighboring cell. That is the all or none law. Let us discuss another important factor of the action potential generation, that is known as the refractory period. A new action potential cannot occur in an excited fiber as long as membrane is still depolarized from the preceding action potential, because no excitatory signal, however strong, can open the sodium ion inactivation gate until original resting membrane potential is reached. It simply means that when the membrane potential is still at plus 35 level, the second stimulus cannot excite the cell or cannot cause another action potentials to be generated, because these plus 35 level or plus 35 millivolt of membrane potential, at that stage the voltage gated sodium ion channels still remain open, and that's why further opening of the voltage gated sodium ion channel cannot occur. So period during which a second action potential cannot be elicited at that same point, even with a stronger stimulus, is known as absolute refractory period. For large myelinated fiber, the absolute refractory period is 1 upon 2500 second. But relative refractory period is a time, it is a half of the absolute refractory period, and it starts after the absolute refractory period is over, when stimuli stronger than the normal can excite nerve fiber. Let us now begin the discussion of our final objective of this presentation, the mechanism of propagation of action potential through myelinated and non-myelinated nerve fiber. So, the stages of propagation of nerve action potential are: an action potential elicited at any one point on an excitable membrane usually excites the adjacent portions of the membrane. A point of an axon suddenly developed increased permeability to sodium ion, and positive charge carried by the sodium ion flow through depolarized membrane. If we look at this diagram, then we can understand that this is a diagram where the plasma membrane is shown, and the inside of the plasma membrane is showing the negatively charged, outside is positively charged. That means the plasma membrane of the axon is polarized. But in this second diagram, if you can see that at this point there occurs some changes in the membrane potential, because the outside of the membrane become negative, inside become positive, and that happens because suddenly a conformational change occurs because of some stimulus, and as a result of that a few sodium ion leaks through the sodium potassium channel leak channels, and positively charged ions when move inside the inside of the membrane at that point become positively charged, and because of that there occurs a local circuit of current flow, where a current flow occurs from the positive to negative. And as a result of that, here you can see that there occurs a spreading of these ions. The positively charged ions then spreads across the membrane on both sides. And this is known as the local circuit of current. And this spreading of local circuit of current, uh, it proceeds on both direction in a neuron. So due to the entry of positively charged sodium ions, a local circuit of current flow occurs between depolarized area and adjacent resting membrane area of the membrane, and that leads to the increase in voltage of the membrane for a distance of 1 to 3 millimeter inside myelinated fibers to above the threshold voltage value. And that is shown in figure B, that a local circuit of current is developed at a certain point, and then the local circuit of current develop, and in C it is shown that the spreading of the depolarization process occurs in both direction. So that's why the sodium channels in new adjacent areas opens and action potential develops in those new areas. And in the figure D, it is shown that the spreading increases further along the axon in both directions. The flow of positive charges continues from this area of generation of action potential ahead and behind this point in the membrane. Thus, the flow of positive charges results in the development of electronic potential in adjacent areas and cause opening of voltage-gated sodium channels. The newly depolarized areas produce local circuit of current flow still further along the membrane, causing progressively more and more depolarization. Thus, the depolarization process travels along the entire extent of the fiber. The transmission of the depolarization process along the nerve fiber is called a nerve impulse. So here you can summarize the events of propagation of action potential. Suppose this is an non-myelinated neuron and this portion is enlarged. This portion of the axon is enlarged. At this point of the axon, suddenly some stimulus is reached, and as a result of that, sodium ion entry occurs to the leaf channels, and that leads to the development of local circuit of current. And because of this, the voltage of the neighboring depolarized areas changes, and the voltage gated sodium channels open, and it leads to the generation of action potential. And this process, in the next diagram, you can see that this process of sodium entry and the generation of action potential spreads, and the repolarization of the membrane behind this action potential also occurs simultaneously. We can explain the conduction of nerve impulse in non-myelinated neuron through this diagram. We can see that the membrane is polarized here. This is the axon membrane, and it is inside of the membrane is negatively charged, outside is positively charged. That's why it is a polarized membrane. But in the next stage, if this point is certain stimulus is applied at this point, sudden sodium ion entry occurs through the sodium potassium link channels, and as a result of that, inside of the membrane become positively charged, and that leads to the local circuit of current development. And this local circuit of current spreads from this point, and it again causes the neighboring or adjacent areas of the membrane to be depolarized. And if the threshold is reached, that means if the plus 15 millivolt increase in membrane potential occurs in the neighboring areas, the action potentials are also generated at these points. And the polarized areas of the membrane now become depolarized. So the depolarization process in this way that spreads in the polarized areas, and behind these depolarized areas, the membrane potential again it becomes the resting membrane potential because repolarization also occurs. But even after that, the sodium ions, when they enter inside cell, they do not go outside. Neither the potassium ion which go from inside of the cell to the outside, they do not come inside. But as we discussed that if the sodium ion concentration increases inside the cell, that may hamper the cellular enzymatic activities. So the sodium ions should go from inside of the cell to the outside to bring about the unequal distribution of ions. Similarly, for the potassium ions which have gone outside should come inside the cell. So this role of bringing about the sodium and potassium ion back into their places is caused by the sodium potassium pump, and that plays a very important role in the neuron to bring back the unequal distribution of ions. So this sodium potassium pump is a very special type of protein channels which can transport the sodium ions and potassium ions against their concentration gradient, and because of that they use the ATP hydrolytic energy, and at a time 3 sodium ions can go outside and 2 potassium ions can come inside to the cell through this sodium potassium pump. Let us now discuss the propagation of action potential in myelinated fiber. In case of the myelinated fiber, the propagation of action potential is called the saltatory or jumping conduction. Similar with the non-myelinated fiber, propagation of action potential depends upon the circular current flow. But myelin is an effective insulator and prevents current flow through it. So depolarization jumps from one node of Renbier to the next with the current sink at the active node. If we look at this diagram, this is the plasma membrane of the axon of a non-myelinated fiber. Here, these yellow portions are the myelin sheaths and which are insulators. And between two myelin sheath fragments, there is the node of Renbier. If an action potential is generated, if a stimulus is achieved, reached in this membrane, then the sodium ion leak channels which are present at this node of Renbier, they open, and as a result of that, sodium ion enter here, and inside of the membrane becomes depolarized, and the local circuit of current develop from this node to the next node, because the myelin sheath present between two neighboring nodes of Renbier is an insulator, and as a result of that, local circuit of current develop between node to node. And when this depolarization process spreads, it can generate an action potential in the next node. And that's why the propagation occurs in a jumping manner from node to node in the non-myelinated fiber. And that is known as the saltatory conduction. So in case of myelinated fiber, the active node electronically depolarize the node ahead. And it is a rapid process in case of the myelinated fiber, since the depolarization process jumps from node to node, and that's why it skips these portions of the myelin sheath or the axon, and that's why the process of propagation is a faster one compared to the non-myelinated fibers. Here in this diagram, it is shown that the sodium ion entry occurs and then action potential is generated, and the spread of depolarization occurs from this node to the next node. Some associated facts regarding the conduction of the uh, action potential in myelinated fiber are that the spread of depolarization or the conduction, the rate of conduction is 100 meter per second. Distribution of ion channels is very important aspect to understand this mechanism of propagation. And if we consider, for example, the sodium ion channel distribution per square micrometer of mammalian myelinated neuron, if we can see that the in the cell body there are 50 to 75 such channels. In the initial segment or axon hillock portion, there are 350 to 500 such channels. But interestingly, 2000 to 12,000 such sodium channels are present at the node of Renbier. And that is very significant because at these nodes of Renbier, the action potentials are generated. Another important component of this conduction is orthodromic and antidromic conduction. The orthodromic conduction simply means that the impulse conduction in when occurs in only one direction, and that may happen from the synaptic junction or receptor towards the axon and up to the termination of the axon. But antidromic conduction means that conduction when occurs in opposite direction in the axon. Here we can explain that if an action potential is generated here, it can propagate on both direction. If it propagates from both direction from this point towards the cell body and also towards the axon, then that will be called as the antidromic conduction. But when it occurs in only one direction from this point towards the axon only, then that is called the orthodromic conduction. Usually, the synapses permit conduction only in one direction. So any antidromic impulses fail to pass first synapse and they encounter and die out at that point. This simply means that if an action potential is generated here, it may travel from this point towards the cell body and also from this point towards this axonic end. But when the propagation of action potential occurs in the towards the cell body, they meet the synapse here. And when they meet synapse here, at that synapses, the action potentials dies out. But if the action potentials travel towards the axonic end, they reach the synapse and then they can pass from this axonic end towards the neighboring cell through the synapse into this cell body. That simply means that the propagation always occurs in only one direction because it occurs from the axon to dendrite and not from the dendrite to axon. And that's why always the orthodromic conduction occurs in case of the neurons. So here is the summary of the saltatory conduction. The myelin sheaths form the insulation, and as a result of that, the sodium ions channels which are present at the node of Ranvier, they function and leads to the depolarization process, generation of action potential, and then the spreading of the depolarization from node to node. And as a result of that, another action potential is generated here at this next node because of the local circuit of current development. So the learning outcome or summary of this presentation are here. We have discussed about the basic structure of myelinated and non-myelinated neuron. We have discussed about the Schwann cell and the oligodendrogliocytes, the special type of cell, the plasma membrane of which makes the myelin sheath around the axon in case of this central nervous system neuron and the outside the central nervous system neurons. We have discussed about the very special junction between the axon of one neuron and the dendrites of the neighboring neuron, which is the synapse. We shall also discuss about the synapse in some other presentation and the synaptic transmission also. We also discussed about the concept of resting membrane potential and measurement of membrane potential with the use of microelectrode and why this membrane potential is negative because it is measured with respect to the inside of the membrane. We also discussed the factors responsible for this unequal distribution of ions and the negative resting membrane potential. And we definitely discussed about the, the diffusion potential, the conductance or the rate of diffusion, then the role of the electrogenic pump, the sodium potassium pump, and also the, the conductance and the electronic potential of the membrane through which the sodium and potassium ion can differentially move across. We also discussed the nerve action potential and its generation in three stages like the depolarization stage, repolarization stage, and hyperpolarization. And to achieve the depolarization, the threshold achievement is most important. We also discussed the ion channels involved in the generation of action potential like the sodium ion, potassium ion, and the sodium potassium pump. We discussed about the characteristics of these channels like the sodium ion channels are having two gates. One is the activation gate, another is the inactivation gate. Inactivation gate and activation gate, they respond to the same change in the voltage or increase in the 15 millivolt membrane potential, but the closing of the inactivation gate for sodium ion occurs slowly. With regard to the voltage gated potassium ion channel, they also respond to the same increase in 15 millivolt membrane potential, but the opening of the activation gate for potassium ion is still further a slow process. And that's why the depolarization process when ends, then only the repolarization process starts vigorously. We also discussed the roles of the sodium potassium pump in bringing back the unequal distribution of ions. in inside and outside of the cell. We discussed the all or none law, where we have discussed that if a, a stimulus is reached in a membrane which can lead to the development of threshold, then only the action potential is generated and it can excite the whole neuron. Otherwise not. We discussed about refractory period, stating that when a particular action potential is being generated, a, a new stimulus with even a stronger stimulus cannot excite the same point of the membrane when one action potential is being generated, and that is known as the refractory period. We discussed finally the mechanism of propagation of action potential through myelinated and non-myelinated nerve fiber, and very importantly, we discussed the local circuit of current flow, which occurs at this point of entry of sodium ion towards the neighboring areas, and which ultimately leads to the development of action potential in the neighboring areas, and that is actually the spread of depolarization. And in case of myelinated neuron, we discussed about the saltatory or jumping mode of conduction of depolarization from node to node. And finally, we discussed about the orthodromic and antidromic conduction in neuron, where the antidromic conduction it cannot cross the synapse, and that's why all the conduction in case of the neuron are basically orthodromic. That is in only one direction. I have used these textbooks for preparation of this presentation. So thank you. Hope this presentation will be helpful for the degree students to understand the basic functional characteristics of the neuron.